At Palmer Lab, after we breed the rats, we send them to the collaborators to do the phenotyping, which is they do the experiments or behavior experiments on those animals. After that, they're gonna send us back a piece of tissue. Well, with the piece of tissue, we'll extract the DNA from it, and then we'll send the DNA to the sequencer, and then we'll get a sequence of each rat from the sequencer, and then do a lot of analysis on them.
We use those DNA sequences to look at differences between animals. So we can look across the entire length of a genome, and one of the main things that we do is we look at particular sites randomly across the genome called single nucleotide polymorphisms, or SNPs, and we can see at each of those sites what variant of DNA does an animal have. Sometimes they might be the same, and sometimes they might be different from each other. And these differences are key to our downstream analyses.
There are also other genetics features we also take a look at. For example, this rat might have this whole genetic sequence deleted, and then this rat might have a whole piece of genetic sequence inserted into it.
Our lab team sequences rats at 0.25x coverage, generating POS-Q files. We then process these sequences through our in-house genotyping pipeline to produce millions of genotypes. Because we use low coverage sequencing as input, and because our pipeline is engineered for scalability, we can genotype thousands of rats simultaneously at a relatively low cost. The resulting genotypes undergo stringent QC procedures. We use machine learning to verify sample identity, and we also incorporate pedigree information as an additional layer of validation to confirm genotyping accuracy.
So once we have run the QC checks on the genotype data, we then publish the raw sequencing data on NCBI's Sequence Read Archive and the genotype data on UC San Diego Library's Digital Collections website. So because we periodically genotype new samples, you'll see different versions of the genotype data on UC San Diego Library. Now, the good thing about getting the data on these public repositories is that researchers can then cite these datasets in their publications.